Discovery Series webinar. Today we'll be exploring new ways to accelerate bioproduct R&D developed at Berkeley Lab. In addition to hosting, we'll also share insights on partnership opportunities with the Agile BioFoundry and with Berkeley Lab. Briefly, I'd like to provide some context on what the Agile BioFoundry is. If you want to go to the next slide, please, Sean. Thanks. The Agile BioFoundry is a consortium of 7 Department of Energy national laboratories focused on developing tools and processes to enable biomanufacturing of diverse products with an emphasis on fuels and chemicals. We partner with academic and industry organizations of all sizes to leverage federally supported research to de-risk bioprocesses towards enabling commercialization of bioproducts. Something to keep in mind during our presentation today is a current call that we have looking for partners for tool development. I'll explain more at the end of the presentations. We're also very privileged today to have 3 presenters who will each share their expert perspectives on their technologies. First up, we'll hear from Chris Petzold, a staff scientist whose focus within ABF is developing mass spec solutions to problems facing biofuel and bioproducts production. Then we'll hear from Hector Garcia Martin, whose work aims to make synthetic biology a more predictive discipline by combining machine learning approaches with mechanistic models, microfluidics, and genetic editing techniques. Last to present will be Eric Sundstrom, whose work addresses critical barriers to process scale-up, intensification, and integration for novel bioconversions, aiming to speed industrial deployment by reducing process costs, development timelines, and scale-up risks. Before we proceed, I'd like to remind you that your microphones will be muted throughout this presentation. And if you have any questions, you can type them in the chat box at the bottom of your screen, and we'll address them during the Q&A session at the end of the webinar. And now I'll hand off to Chris.
Thanks, Grace. Let me share, and we can get started. Good morning, everyone. My name is Chris Petzold, and I will tell you a little bit about the Agile BioFoundry's automated pipeline for strain analysis. And I'm, like Grace said, I'm part of the test team. So Grace gave you a nice kind of overview of what the ABF is focused on and the developments that we do. And within the ABF, we do additionally demonstration projects to kind of show the tools that have been developed, the capabilities that the ABF has, and really try and move the bioeconomy forward. And I'm going to highlight a couple of them today and focus mainly on one of them. You can see that they're separated in two cases, by organism and by the target molecule that's being produced. In one case, it's the host is Pseudomonas butida, and the target molecule is mucinate. And in the other case, Rhodosporidium torreoides is the host, and it's focused on producing lipid molecules. Now, in each of these cases, there are different needs and different areas of focus for these experiments in the experimental design. In the Pseudomonas case, how to screen pooled genomic and genome scale libraries for mucinate production is a key aspect of what the research is moving towards. And in order to do that, we really want to be able to connect the genotype with the phenotype that we observe. Now, some of the examples are we have RB TNSeq libraries, CRISPR-I knockdown libraries, as well as gene overexpression libraries that can be screened and assayed. And while there are established methods for analyzing these, a lot of them require either a growth-based, selection-based methods such as AL, automated lab evolution, or screening technologies where there's already an existing biosensor that enables droplet or FACS analyses. Now, in the case of mucinate, while there are biosensors and other technologies out there, there are a lot of cases where you're just not able to use existing technologies. And so we need a fast, non-growth-based method of phenotyping and genotyping these strains that are being created. Likewise, in the Rhodosporidium case, being able to screen large numbers of transformants to ensure that the chromosomal modifications are in the correct place, as well as performing the correct function, are really key. And Rhodosporidium has a unique cell wall composition and a high GC content, which makes colony PCR difficult. And so developing workflows that enable our technologies and our demonstration projects is really kind of where we're going with a lot of the tools development at the ABF. Now, to give you an example of the ABF strain analysis workflow, it generally starts at a culture transformation step where DNA is transformed with a host of interest, and it's then subsequently plated on agar plates for initial colony growth. At that point, the colonies need to be picked, and in all these cases that I demonstrate here, they're all commonly done manually in industrial labs and academic labs and at all levels. But being able to do that in high throughput is really the key. The culture growth step there on the right is actually a very highly variable portion of the process, where growth in 96 well plates or biolector or other bioreactor formats can often be dictated by the experimental need of the host or the target molecule. And like I was saying on the previous slide, being able to do product extraction and assay them, it can be highly variable and is often a rate limiting step. So what I'm going to talk about today is an example study of the Pseudomonas butida for mucinate production, wherein the strains are generated, they're grown, and then we do the following analyses. We do product extraction and product analysis to see how much mucinate is being produced. We do proteomic data acquisition to assay what the proteome components are within those strains, and then we also do genotyping so that we can utilize that genotypic knowledge for downstream learn activities. So focusing on the colony picking through production culture step, this is typically the initial stage, wherein colonies are picked via a QPix automated colony picking system into 96 well plates. These 96 well plates are then handled in step 2, where they're grown overnight to produce the initial cultures that we're going to use for the production growth assays. Now, in step 3, this is an area that can be highly variable depending on your needs. Now, in this case, I'm demonstrating that the nunc plates are being generated in triplicate in deep well plates for production growth, but that can also involve multiple passages into different media conditions or different growth conditions depending on the host and or the need. And likewise, within that step, we are sampling part of those initial cultures into 384 well plates for genotyping purposes. And then we also do stocks in case we want to return to those cultures at a later time. And lastly, once the production is proceeded for the appropriate length of time, we do sampling for both the proteomic samples of cell cultures, and in this case, for mucinate, we are sampling the supernatant of the cultures for the rapid fire assay. And this rapid fire assay is one of the key aspects of being able to match the throughput that we see from the colony picking and strain generation stage in the actual testing and analysis stage. This is again a highly variable step in the process depending on your needs for your product. In this case, mucinate is secreted into the media, and so being able to just simply sample the supernatant, dilute with the appropriate solvents, enables the actual assay to proceed. But in some cases, if you're looking for intracellular metabolites or you're looking for lipid molecules where you need to fully lyse the cells, different extraction protocols need to be established. And we have a variety of them in automated formats at the ABF. Likewise, one of the main advantages of the mucinate assay is the throughput. Now, being able to analyze targeted molecules on the order of 4 to 5 samples per minute is very much higher than traditional methods such as liquid chromatography or gas chromatography systems. This enables us to compete, or maybe not compete, but at least complement the throughput that can be generated at the transformation and library stage, where we can get thousands of samples analyzed per day. Now, we validated these rapid fire assays with the more traditional methods to ensure that the quantification that we get from these methods are accurate and representative of the cultures that we see. Now, in addition to just using this workflow as a screening tool where you're picking the winners and moving on and only analyzing those, we want to enable more deeper levels of learning activities where we can really learn more about the system. And these techniques you'll learn more from Hector in a few minutes. But to aid that, we have developed high throughput automated proteomic sample preparation protocols to take the cell pellets and digest them all the way down to peptides for LC-MS analysis. And because the traditional proteomic assays are often time-consuming and work-intensive, we've really focused on being able to do this in high throughput, where we can analyze 200 proteomic samples a day and be able to quantify over 1,000 proteins from each of those samples. This provides a rich data set for strain validation, just analysis of stress response and other aspects of the host as it's undergoing its production assays. And we can implement that throughout the time course of the growth. We can implement it as an endpoint, and it really helps to inform what else is going on and feed in more advanced machine learning technologies that you'll hear about later. But it also, we've developed automated methods to provide analysis of the proteomic samples in a high throughput so that it's a timely way of getting the data to the users and the collaborators. But ultimately, we really want to be able to identify the genotype of these strains and see what genomically is matching up to the phenotypes. And we've developed colony PCR methods, as well as Illumina library preparation and MiSeq sequencing methods in high throughput and in automated fashion to really enable the ability to get through the entire process in a timeline that actually is actionable and can be moved forward. Now, in some cases, this involves just simply sequencing the barcodes of a TNSeq library, or it can be involve more involved sequencing for CRISPR-I or gene overexpression libraries. So just to give you an example of our application list for an RBTNSeq library, we arrayed and phenotyped 16 plates colonies of approximately 1,500 colonies with control strains on each plate. And as you can see on the right, an example of what we would expect to see in terms of the mucinate production, where there's a lot of production that didn't change very much across the different TNSeq library strains. And then there's some winners and then there's some losers. And as you can see from this this data profile, we can see that in this case, there were quite a few of the strains that actually produced a significantly more mucinate under the conditions that we screened. And so being able to feed this information into advanced learning methods is really a way that the ABF is providing workflows across the needs of bioengineering and the bioeconomy. So just to bring you back to the workflow that I started with, there are multiple user on-ramps for these methods with the ABF. Users can bring their own host and bring their own DNA for transformation and procedure through the process. They can bring transformed cells that are already ready for colony picking, or they can bring seed cultures that are ready and prepared for high throughput production growth. And lastly, if collaborators want to bring existing extracts that are ready for high throughput analysis with the rapid fire or other techniques, the ABF is well suited to work with you in those cases. And with that, I'll acknowledge the people who helped out with this and let you hear from Hector about what we can do with all this data. Thanks.
Okay. Hello, everyone. And yeah, so now that Chris and ADF can produce lots, thousands of data points for you, what do you do with that? And that's what I'm trying to show you. See, can you see my screen?
Yes, but it's not in the full view. It's in the presenter view mode. There we go. It's probably going to be better.
Let's see. Let me try that again. Sorry. Good now? That works?
Looks good. Good. Yep.
Okay. Thank you very much. Okay, well, so yeah, now that we have that AVF can produce thousands of data points for you or tens of thousands, what do you do with that, right? Maybe standard analysis using Excel sheets and biological intuition has a limit and it can't really deal with so much data. So I'll give you an example of one tool that we developed, the automated recommendation tool, that is able to leverage that data to guide the metabolic engineering or the synthetic biology process effectively. And this is just one of several tools that we have developed in my group as part of the ABF, and you can go to qmm.lbl.gov to see many other tools and examples that can be useful to you. And well, the reason that we do all of this is that, well, synthetic biology can really enable a circular bioeconomy and it's produced some interesting products already, but it's too slow, right? And the reason is that we don't have the predictive power that is needed. As the Tim Garnett said at some point, our ability to engineer predictable outcomes from biological systems remains infantile. We can make the changes we intend, particularly with the new genetic editing tools based on CRISPR and so on, but the effect on cell biology is generally unexpected. And we think we can fix that with machine learning and with the predictive power that machine learning can provide. But that, in order to feed the amount of high quality data that machine learning needs to work properly, we need automation. And I'll give you an example of what that means. Now, A.R.T is a machine learning tool that can provide the predictive power needed. And what it does is just takes an input and a response. And this could be many things. A.R.T is a general tool that can be used for a variety of different tasks. Could be as input protein concentration and then the amount of biofuel you're producing, or the promoter combination in your DNA and the productivity, or it could be the media components concentration for each of the media components and the flavulin titer. And then it takes all this data, it learns from that, and it creates a predictive model that tells you, well, this protein profile has 10% of chance of producing 10 milligrams per liter, 20% chance of producing 50 milligrams per liter, and so on. So it gives you a probabilistic model. And from that, it gives you recommendations. So decrease this protein 1 to half, double this protein 2 to double to get an increased production. And it gives you those recommendations. And you can see some examples in a couple of publications, the initial A.R.T paper and an application of that for a promoter combination decision. Now, A.R.T has been designed for synthetic biology needs, first because it can work with very few instances. And you can see that in the paper, we've been able to obtain good results or acceptable results with as low as 27 instances, which is something very, very rare in AI, where people are focused on the high end of instances of training data. It does provide uncertainty quantification, so you can tell whether you can trust the recommendation of the prediction, depending on how much uncertainty is associated with that. If you have like a wide probability distribution, you can't trust it too much. If you have a very narrow one, then you can trust the recommendations quite well. And then finally, it doesn't just produce predictions, but recommendations. It really goes to the gap of between learn and design very efficiently, and it tells you, well, change this protein or try this promoter combination and so on. So it does provide recommendations that are actionable and you can use to really mind this gap between learn and design. I will now give you an example because I think this is best seen and appreciated through an example in which we're optimizing media. We were trying to optimize or increase the flavulin titer, and we have 15 media components, which we changed 12 systematically over 2 orders of magnitude to see if that made an impact on flavulin. And from that, again, we got several instances around, you'll see around 15 per TVTL cycle, and we did around 5-6 cycles, around 60 of those, and we're able to predict response. And that created a predictive model that that predicted that this media design has a certain 10% chance of producing 10 milligrams per liter. And then we did recommendations that said, decrease this component 1 and increases component 2 and so on. In order to provide enough data for R to work well, we created a semi-automated pipeline that allowed us to create new media in a high throughput manner and without errors, which is important. We had a biomech, a liquid handler that, given a media composition, was able to put it together. Then we did the fermentation in a biolector with where we supplied the organism, antibiotic inducer, and so on. We assayed production through fluorescence. Then we gathered this data into the experiment data depot over here. And then ART pulled the data from the experiment data depot, made a recommendation for new media, and then it passed it to a Jupyter notebook that took those concentrations recommended, and then it figured out what are the robot handler steps that it has to do to create those media recommendations. What we did see in the first time we tried it, we had initial DBTL0 to check reliability, and then 2 DBTL1 and 2 to with around 15 instances each. And then we trained ART with this data, and we saw that the predictions versus the observations were not incredibly good. They had an R-square of 0.32, but they were at least not random. And what was interesting is that in the next DBTL3 cycle, we were able to use the recommendations to increase production by around 148%, which was quite unexpected. We didn't expect media to have such an impact in production at the very end. We did this 3 times, and we obtained equivalent results. We did it, this is the original one, where we did 5 cycles, 5 DBTL cycles in total. Then we did it again with exactly the same conditions. And in this case, we had to wait until DBTL5 to get an improvement in flavulin, and it was around 170% in titer. And then we tried to do it with yield, and we got an increase of around 300 in the third DBTL cycle. So this seems to work, and work quite well, and in a repeatable fashion, with of course some stochasticity. But what was interesting is they provide really surprising results, in the sense that sodium chloride was the most important media component to explain production. So this is the SHAP value. That means that this is the impact in the model. That means that if it has a high value, if sodium chloride is high, it impacts the production positively, and if it has low value, it impacts the production negatively. And what was really surprising is that sodium chloride was the most important of that. And in fact, the optimization took us to the limit of almost to the limit of what Pseudomonas butida can really withstand. So that's just an example of the surprising results and the optimization and that machine learning can bring to your project. ART is being used in a variety of projects. As I said, it's a general tool that can be applied to different problems. We're using it to give targets for CRISPR-I to downregulate, to increase sustainable aviation production. And these are each of the DBTL cycles, when you can see the isoprene titer increase significantly. We're using it for designing polyketide synthesis and creating what we think are the first quantitative predictions of the production of polyketide synthesis. And we're also using to optimize electroporation conditions in a microfluidic survival lab. And we're also finally using to predict the scale-up, how to go from biolacto to amber in a predictable fashion. But these are things that we're working on right now, and I hope to publish very soon. If you're interested in ART, there is a website with tutorials and explanation of ART and tutorials, and even a publication that explains how to use it. And if you're interested in other tools beyond ART, you can actually check. You can also check qm.ldl.gov and get more information. And that's it. Thank you very much for your attention.
Great. Is that working for everyone?
Looks good.
Great. So I'm Eric Sundstrom. I'm a staff scientist at Berkeley Lab. I lead the fermentation and recovery team at ABPDU, the Advanced Biofuels and Bioproducts Process Development Unit. We are a very complementary capability with the Agile BioFoundry. So our specialty is really process development and process scale up. And I think kind of a nice way to frame it is to think about the concept of frozen here. The concept of the scale-up valley of death. So many of you have probably heard of this. Essentially, there's a lot of government and university activity at low technology readiness levels, typically the types of projects that would be funded by NSF or early stage Department of Energy funding. And then there's, of course, a lot of interest in the private sector in adopting and commercializing these new technologies. But there's this sort of gap in investment where it's not necessarily as interesting to the basic researchers. This requires expensive capabilities, things that may not be off the shelf, but the technologies haven't yet been sufficiently de-risked for the private sector to be putting money in. And so ABPDU was really founded to step into that gap and help catalyze that de-risking step to enable technologies to move to the private sector. And I think we've been quite successful in that regard over the years. We've been around, I think, about 12 years now. ABPDU has had 85 industrial partnerships over that time. Primarily, these are small companies looking to access our capabilities, but that can also include larger companies that have specific needs that aren't met by their facilities or that are looking to access other resources in the national lab system. We also work extensively with academic and national laboratory partnerships, including many consortia, such as the Agile BioFoundry. The key capabilities at ABPDU are primarily around bioprocess scale-up, process integration, process intensification. So we have high throughput fermentation platforms like the Amber 250 system, which is a fully automated high throughput fermentation screening platform. Our capabilities go up to 300-liter pilot or pre-pilot, which can be very useful for assessing scalability and also for generating larger quantities of product. And then we also have a gas fermentation capability. This is a newer capability, and that's what I'm going to be focusing on today. And the reason we've invested in this is that there is a whole host of feedstocks that are really ripe for bioconversion, and there's a bit of a gap in terms of capability that's out there to access those feedstocks. So this would include things like electrochemical feedstocks, particularly green hydrogen, which is really an up-and-coming technology that's seeing a lot of investment and a rapid decrease in cost, especially coupled with renewable energy, which is similarly seeing a major decrease in cost. It also includes carbon capture and CO2 utilization, anaerobic digestion, which is a pretty robust source of renewable methane gas, biomass gasification, which is similarly a way of converting biomass into these C1 feedstocks, and then also a variety of industrial waste gases like flared natural gas, steel mill off gas. There's a lot of C1 feedstock out there that can be converted biologically, and there's a lot of groups interested in this space. So we have groups looking at production of very low cost things such as foods, medium cost items such as polymers and biochemicals, and we even have groups looking in the food space. We've worked with a group looking at making chocolate, for instance, from these electrochemical feedstocks. So it really covers a broad space. And at ABPDU, when we did some industry outreach in this space and talked to a lot of the practitioners, there was a pretty clear consensus that there is a gap as far as scale up that is even worse for gas fermentation than it is for typical sugar-based fermentation processes. The reasons for this would be that the space is fairly nascent, so there's a lack of effective scale down capability that can mimic what you will see at large scale. So a serum bottle is kind of the gold standard for what people are doing in the lab bench. And it really does not replicate what they see at large scale in terms of gas partial pressure, total gas supply, biomass density that you can achieve, pH control. All of the different controls you can put on the bioreactor are just not the same. There are barriers to operating these in a safe fashion, both at the bench and at large scale. Particularly flammability comes up a lot, but also gas toxicity if you're working with carbon monoxide. And then these systems often work under pressure, which creates additional barriers to getting on that equipment. And there's really no established best practices. there's very few off-the-shelf pieces of equipment. So our goal is to kind of step into that space and provide a resource that's accessible to people who are just entering and want to be able to exercise their process under industrially relevant conditions. So we've invested in the capability you see here. The first to come online was our 8x500 mil bioreactor block. These are HCl bioreactors that are individually pressure controlled and fully stainless steel and enclosed in a fume hood. And so what that allows us to do is operate with essentially any mix of gases in a safe and secure fashion, and also to put pressure on the system that can mimic pressurized systems or even hydrostatic pressure at depth that you'd see in a very large bioreactor. Our more recent capability to come online is the 20-liter bioreactor. We just recently installed our gas supply system for that. We have a hydrogen generator that's coming online so that we can move away from gas cylinders and finite gas supplies and move towards being able to run with large volumes of gas at scale. We can run with any combination here of hydrogen, oxygen, CO2, CO, methane, ethane, nitrogen. We'll have to do safety onboarding for those processes, but we've got them all onboarded and tested out. We can do back pressure control up to 5 bar, which can drive very high gas transfer and also subject strains to conditions that you're likely to see during scale up. We also have off-gas analysis set up. So we have an Agilent micro GC and we have a thermoprocess mass spec so that we can close mass balance around these fermentations. So just to give a flavor of some of the different projects where we've applied these, first one I'll talk about is industry collaboration that we had with Checkerspot. This was run through Biomade along with UC Davis. And the idea here was actually not gas fermentation, which made it a really nice commissioning exercise for us. It was to look at pressurized fermentation and how pressure affects scale up of bioprocesses, especially the Checkerspot heterotrophic bioprocesses for production of fats and oils. So what we did here is that we ran a series of fermentations in the small scale bioreactors at low pressure, at high pressure, and then we ran comparable conditions in the 300-liter bioreactor, again, varying the back pressure and also the feeding parameters to mimic what they'll see at small and large scale. And we were able to resolve some initial scale-up issues and get that variation down below 10% between the small and large-scale bioreactors, which helped them resolve a hurdle towards scale-up to 15,000-liter and commercial deployment. Just in January of this year, they actually ended up releasing the product that we were working on here. This is a new infant formula. So they're tailoring fats to match those that are found in human breast milk. We've also been working with a number of other companies, Cersei Bioscience, Industrial Microbes, Winfield Bio is our latest partner here. And we're excited to continue to work with more companies in this space. Also talk about some of our more basic fundamental research. So here we're supporting 2 projects from the Energy Biosciences Institute. On the left here, we're working with thermophilic archaea, acidothermophilic archaea, and we're looking at C1 conversion in these organisms to see if they can be good hosts for production of thermotolerant enzymes, for instance, or other kinds of thermochemical processes. And we're able to co-feed formate and hydrogen and achieve biomass densities and formate conversion that otherwise would be inaccessible. On the right, we're working with Cuprovitus necator, which is kind of the workforce host for conversion of CO2 and hydrogen. And what we're looking at here is the CO2 uptake pathway and whether we can drive CO2 conversion from air. So could this organism essentially serve as a carbon concentration mechanism to utilize CO2 directly from air? And this is an experiment. It worked. We were able to see growth here just from hydrogen and air. And it also kind of emphasized the value of having this system because you cannot do this experiment in a bottle. You need that continuous supply of hydrogen and air. We need this scaled down system in order to actually determine whether these bugs can hit that metric. Because if you try and do this in a bottle, they'll immediately run out of carbon dioxide. Another project we've been working on here, this is conversion of CO2 and electricity to fuels. This is a Bioenergy Technologies Office project where we're looking at conversion of CO2 to carbon monoxide. and then aerobic fermentation of carbon monoxide to produce fuels and chemicals, which allows production of long-chain fuels and chemicals that are essentially ready to go as sustainable aviation fuel. So the typical path we've seen in this space has been to ferment syngas to shorter chain intermediates and then assemble those catalytically into longer chain fuels. We've shown here that it's possible to aerobically convert carbon monoxide directly at very high rates and to generate very high densities of biomass via this route. So we were able to get 27 gram per liter dry cell weight from just CO and hydrogen feedstock, which is 200 times higher than what had been reported in the literature without access to this kind of feedback-based control on the gas supply, pressurization, continuous gas supply, all the things that are enabled by this kind of equipment investment. We were able to show pretty rapid rates of CO conversion in that system. And the last one I'll touch on is electrochemical integration. So we've been working a fair bit as well on different configurations where we could directly integrate generation of electrochemical gaseous feedstocks with conversion. That simplifies the process and also allows for direct dispersion of the gases into the system. So this is an example where we actually did water splitting electrolysis in a fermentation vessel and used that to generate a expression of a heterologous product, actually the same molecule that Hector was just talking about, flaviolin. So we can produce it in this tank from just electrons and CO2 in a single vessel and grow a genetically engineered strain of Cucurbita nectar to express production of that product, which would allow you to produce these things from electricity essentially anywhere you have access to a CO2 source. So this is just kind of a flavor of the things we can do, where the whole purpose of these capabilities is to support collaboration. So if any of this kind of jogs some potential application in your mind, I'm very happy to discuss further.
All right, thank you, Eric, and thank you everyone for your attention. I'm just gonna go ahead and share my screen. Okay. All right. So before we move on to the Q&A session and hear a little bit more from our presenters, go ahead and keep adding those questions to the chat. But I'll take a few moments here to discuss how businesses and other organizations can partner with Berkeley Lab and with the Agile BioFoundry. I hope I'll be able to provide you an opportunity to understand how our strategic partnerships and technology transfer work within the framework of a national lab and how they can be leveraged for commercial success. So my role at Berkeley Lab and with the Agile BioFoundry is really to support our scientists in getting their technologies into the marketplace and to foster these partnerships and collaborative research. So at Berkeley Lab, there's several ways to kind of leverage these capabilities and work with us. Firstly, our intellectual property office is responsible for patent and software licenses. This office also handles agreements such as NDAs and MTAs that are typically needed prior to you taking on the license of our IP. We provide all matters relating to IP, including IP management with other institutions and labs. And IP also works closely with our strategic partnership office on IP terms, and they handle the various agreements such as user agreements, strategic partnership projects, or cooperative research and development agreements. So what are those? This is a DOE chart that spells out the specific terms regarding IP and data rights you get under these different types of agreements. So first up here is a CRADA, a cooperative research and development agreement. A CRADA anticipates both partners work together under an ownership follows inventorship type model. So typically these relationships will last several years and the scope will have tasks for both parties. You can work on-site alongside our scientists under a CRADA, and in the IP terms here, the lab will take title to our subject inventions, but the partner has an option to negotiate up to an exclusive license in a pre-negotiated field of use. Data can also be CRADA protected information held for 5 years, and our background IP is provided to the participant at no cost for the duration of the project. Second type of agreement here is a strategic partnership project, or SPP, used to be known as work for others, if you recognize that. And this is a classic sponsored research project where an external partner funds our scientists to do a specific scope of work on their behalf at essentially the same cost it costs us to do the work, so it's full cost recovery. If the sponsor is a US company using non-federal funds, the subject inventions can belong to the partner. This chart also shows user agreements at the bottom here, and we do use these at Berkeley Lab, but they're not very Agile BioFoundry relevant. So I'm happy to answer questions about user agreement opportunities and where else at Berkeley Lab you might see these pop up. If there's anything that comes up, feel free to drop that in the chat. Okay, so specific to Agile BioFoundry, both at Berkeley Lab and utilizing those other 7 labs capabilities. And we have some partnership information available at the site on this QR code. And I also saw that Sean dropped the link in the chat, so you can follow that. And at this link, you'll be able to view a created template. So that's the Cooperative Research and Development Agreement. And this template is the form that we use for all of our projects that utilize DOE funds. So this form is non-negotiable if you do have that DOE funding. And the point of this is to aim to speed contracting so we can actually get to work faster. I think we all know contracting can be a little bit of a barrier. And when you're working with multiple national labs, this is a great way to get that done quickly. So you can go ahead on that link and review that form if that's interesting to you. And then I'd like to highlight one opportunity that I mentioned at the start that we have available right now that's relevant, I think, to what you heard today. So this is our Strategic Core Industry Partnerships, or SCIPs as we call them. In these projects, our industry partners will provide up to 20 or at least 20% cost share in addition to industry perspective and expertise and collaboration. So those tools you heard about today are available for these partnerships, as are several others, and you can read about all the available opportunities on that partnership site, as well as a skip for these core for biochemicals and biofuels that I mentioned at the start of today's presentation. You'll also find on this Work With Us site a list of our licensable ABF technologies, both from Berkeley Lab and from our partner labs, including those that you heard about today, and those links will take you to more information. And you can also just email me, and I can help you through identifying the IP you're interested in, all the way through executing a license agreement, or direct you through our partnership opportunities. So I'll end here. I've already seen some great questions in the chat, and we can start addressing them shortly. And please continue to submit those queries as we proceed, and we'll aim to cover as many as possible. If you have more questions, something comes up later, here's our contact information for all the presenters. And then at the bottom of the screen, again, you'll see the link to the ABF website, as well as our link to IPO at Berkeley Lab, where you'll see all of Berkeley Lab's licensable technologies. Okay, so now, oops, I did not mean to stop screen sharing. I'll throw that back up, but it's Q&A time. So I'm going to read through a couple questions. Let's see what has been answered already in the chat. All right, I think it'll be for Eric up first. It looks like this question to Chris got answered. So Eric, how do you clean the bioreactor after one specific experiment?
Yeah, good question. So our bioreactors, essentially anything at the 10-liter scale and below, is autoclavable. So we can fully autoclave everything. Anything at the 20-liter scale and above is steam in place. So all of those have integrated steam in place systems that can fully sterilize. So we haven't ever actually seen any kind of issues with contamination from one run to the next, for instance.
Thanks, Eric. Okay, also for Eric, is it possible to have mycelia production in such bioreactors?
Yes. So we have grown in our sort of heterotrophic bioreactor systems, we have grown filamentous fungi, and we haven't seen any specific issues there, but you'd have to make sure that everything's tested out so that there's no issues with shear force, especially. We have not done it in the gas fermentation bioreactors. I don't see any reason why it wouldn't work. It's just a question of what people bring to us. So happy to discuss further.
Okay, one more for you while you're on. Oh, maybe this is a follow-up. Even if it contains solids, is it autoclavable? I assume that's to you, Eric. Follow up to that question you just answered.
I guess would depend on the application. So as long as it can be, there's no problem autoclaving solids, I guess. The question is whether how the solids would respond to autoclaving. So we do have some feedstocks like hydrolysates that are challenging in that way, where you could get caramelization or something if you were to actually autoclave it. Those are trickier to deal with. You can sterilize them other ways, or if not sterilize them, you can pasteurize them, for instance. So you have to get creative sometimes.
Okay, one more. On your slide about water or electrolysis in the fermentation broth, do you observe poisoning of your electrodes due to the presence of several biomolecules in the fermentation broth?
Yeah, that's a great question. So I wouldn't necessarily say poisoning, but we do have to, when we do these integration experiments, we do have to change our broth to eliminate any ions that are going to create issues. So that would include things like chloride that may result in chlorine formation or may create side reactions that create toxicity. We did actually see in that particular experiment that the electrolyzer was actually active in chemically altering the product. So that was something that's unexpected, but potentially down the line, you could see it. And I do think you do have to pay close attention if you're going to integrate those unit operations to very long time scales to make sure that your media, your product, everything is compatible.
A follow-up on that one, are there proteins in your broth that lead to poisoning?
No, I don't know if protein specifically. I mean, but over time, I think it's potentially that there could be, if there's cell lysis, for instance, we could get proteins in the broth, and you could get more complexity in the broth. We didn't see anything like that in the timescales that we're operating. I do think if you ran them for very long timescales, you'd start to see these kinds of failure modes.
Okay. Any employment of single-use technology bioreactors or other process equipment, wave-style bioreactors?
Yeah, so the Amber 250 system is single use, and it has been used for things such as cell culture, where single use is typically deployed. We don't have any single use capability above that scale. Typically, those are more common in biopharmaceutical applications. It is something we've thought about. We haven't really moved into cell culture. That's also something we've thought about. I think wave style may be more popular in those applications where it's low shear. So in general, there's a pretty robust contract research environment for biopharmaceuticals, and we haven't seen it as really a core part of our DOE mission. But it's definitely something that we're open to. It's something we've thought about.
Okay, thanks, Eric. So question, I'm not sure which of our presenters might be able to answer this, but a question about biodegradable batteries. So this company is fairly new and looking to raise funds and get their battery safety tested. Is this something that the ABF can assist with? I'm not sure if any of our presenters want to take that, or Nathan, even if you want to jump in and give some comments.
I'm happy to chime in. Grace, specifically, what did you want me to speak to? Sorry.
That's fine. So the question is from a company making biodegradable batteries.
Oh, yeah.
And they're looking to get their batteries safety tested. I'm not sure if that's something that the ABF might be able to assist with or not.
I don't think that's going to be something that the Agile BioFoundry is directly going to be set up to give you some support on. But I think in the chat, Sean did put in a link to lab partnering services. So that might be a good place to start. And the other, just the general websites for the national labs, including Berkeley Lab, might be another set of resources. But the Agile BioFoundry specifically probably isn't set up for doing battery safety testing.
Yeah, so you might check out this ipo.lbl.gov link as well and give it a look, see if there's anything there that might be relevant. Okay, a couple more have come in. I think this will be for you, Eric. Again, is it economically feasible to use a single-use bioreactor?
Yeah, so I guess the answer to that is it depends. For process development, certainly, I think the labor savings at small scale are higher than the cost of the bioreactors. In a production setting, I do think we've seen the pharmaceutical or the biopharmaceutical industry move towards single-use and plastic bioreactors because they have very, very stringent sterility requirements. So if your product is very high value and produced at relatively smaller bioreactor scales, I think it's possible, but for any kind of fuels or chemicals or industrial type products, likely not, unless somebody comes out with some kind of radically new design that would lower the cost.
Okay. And then also for gas fermentation, can you describe your method for determining mass balance in a little more detail?
Yeah, that's a great question. So for gas fermentation, we have separate mass flow controllers for the flammables, for the oxygen, for the carbon source, and then for an inert gas. So we know the rates that we're putting these gases in. We also have a flow meter on the back end, so we know how much gas is coming out. We can also kind of calibrate that upstream of the run to make sure that our flow meters are matching what we think they're putting out. Then on the very back end, we also have, for our smaller scale system, an Agilent Micro GC, which has a manifold that can cycle between the 8 bioreactors. So it can continuously measure every couple minutes what's coming out the back end. We can combine that with the flow meter, and we can also combine that with the inert gas feed. So sometimes we'll add a little bit of argon or nitrogen as a tracer, and we can use the ratio of the other gases in the off gas to that inert gas to make sure that we're aligned on the gas feed rates. So it's more complicated than you would think to get good mass balance, but we have a good handle on it now.
Okay, so one might actually be for Sean here. When is the bioenergy liftoff report coming out? I'm not sure if anybody knows.
Thanks, Grace. Yeah, I was actually looking at that question right now in the chat, and to be determined, but I will make sure that I provide a link to the liftoff reports on this site. And if you'd like to find out more about the liftoff reports and when they're released, you can subscribe. I'll put the link into the chat right now so that you stay up to date on all of the latest reports.
Okay. And Hector, maybe you'll want to take this one. How can generative AI like ChatGPT improve the outcomes of ABF efforts?
Yeah, good question. So there's a variety of places where things like generative AI could be of interest. An obvious one is literature reviews, see what's been there in the past, has been used, like for example, in media components, for example, or gene knockouts or things that have worked in the past. Another place where it could work out is by creating DNA large language models. That's something that we have been collaborating with people from Argonne and other places to create large language models that really utilize all the 700 and 540 terabytes of DNA sequencing that GAGI provides per year to make sure that we can use that data with phenotypic data to create foundational models that help us predict things better. So those are the two things that I can think of right now. I think that, of all the AI things out there, generative models are probably the least useful for us because there isn't a lot of data. We don't have the density of data to make their predictive capabilities really useful, right? For that, what we need to do is collect more of that data in a more systematic way and then make it available to many of these models. But again, we don't have my feeling when the times that we will use these kind of approaches, there is not enough density of scientific data to create really useful recommendations. But we keep working on that, and we can create bigger databases that can be leveraged by LLMs, such as ChatGPT or Claude or any of these, to really try to do things to suggest things that or do recommendations or do predictions that can help the researcher.
All right, thanks, Hector. It looks like that is going to wrap up our Q&A session. So if we didn't get to your question, although I think we hit all of them, we can follow up with you by email within the next week or so. And again, go ahead and jot down these email addresses or go to our site if you have any other questions. And thank you again for your participation in today's webinar. Your engagement and interest are greatly appreciated, and we look forward to welcoming you to future sessions.